All right. Thanks for making the full rotation, guys. I'm going to ask you just while we talk if you could fill out our sign-in sheet. It will allow us to keep doing work with cover crops. So thank you. And Jen, take it away.
Great. Yeah, thank you everybody for coming. So my sample is a little bit pathetic now. I keep messing with it a little bit. But yesterday we talked about all these like awesome hot spots in biological life and where the zone of influence is happening. And it starts. It doesn't start anywhere. It's all connected. But we can start at the top with the litter layer, and it's protecting the soil from erosion. It's moderating temperature. It's allowing, minimizing evaporation to make every drop count that hits the ground. And then that's just facilitating and feeding this nice, this one isn't so much, but this nice plant here. And then you can actually still see, even though I've thrown it on the ground and broken it apart, this aggregation that's happening right around the roots, like what Gabe was talking about earlier. The fungi and the bacteria are there, and they're releasing all these sticky substances, and their hyphae and the roots themselves are enmeshing like a hairnet, right? And they're holding on to that, and they're building organic matter. They're not only building organic matter, but they're creating a whole unique environment for this plant to be as healthy as it can be. And so with a diverse suite of microbes and other organisms that are eating those microbes, it's going to help this plant ward off different stresses, drought, too much rain. Excuse me? Erosion, absolutely, yeah. And also any predation or pathogens that might be out there trying to attack it. They're going to come and swarm the roots. They're going to send signals above ground. And in this area, there must be, tons and tons of different insects, beneficials that are here because it is such a diverse area right here. And the plant actually releases these volatile chemicals, these smoke signals in the air, SOS, if they are being attacked. But they won't, the organisms that can help this plant ward off that pathogen or that stress won't be there if we don't provide that diversity. It has to exist. Cover crops are also amazing. A lot of the nutrients that we apply year after year with our cash crops are leaching down below the root surface, the root zone. And there's just like a whole mine of nutrients that is below that area. These roots, when we grow cover crops, are going to reach down below that area. They're going to suck up those nutrients, bring it back up to the plant, keep it in the roots, build it in the vegetation. And then we're not harvesting that. We're growing for the soil, right? One for us, one for the soil. And then that's going to break down, and it's going to recycle, and all those nutrients, it's going to stimulate organic matter formation. It's going to stimulate a whole suite of different organisms. And the more diverse, the more chances you're going to have the good guys there to fight off the bad guys, the bad nematodes or the bad fungi that might be resting in your soil. So it's kind of like natural biological population control if you create the right environment for it, right?
Probiotics.
Exactly. So I'll kind of pause there for a second and see what kind of questions you have. I'm sure you've just learned so much from the other presenters. Yes, sir.
I'm curious if you guys have been studying cover crops as biofumigants and if that affects the composition of the culture in the soil as well.
Well, do you want to just mention the sun hemp thing?
Well, I know that people at UH are studying mustard as an nematicidal biofumigant, and also that the sun hemp here does excrete a compound that is nematicidal as well. So it kills particularly root knot nematodes, which is helpful for a lot of people. Yeah. I'm not sure if there's research that is relevant on that on the mainland or not.
Oh, absolutely. Yeah, Bob talked about replant disease at the end of our thing yesterday. And that's when you build up a population of the bad guys. And there's a lot of research that with mustard actually that has a much longer suppression of the different organisms. In this case, it was a nematode that was attacking the apple trees. And so they tested it with fumigation, and yeah, it knocks it down. But guess what? That next year, the nematode that was causing all the problems, their populations exploded in the area that was fumigated. But the one that had received the mustard seed meal treatment, those populations were kept low, and then their yield over the 5-year period were actually significantly higher with this organic amendment. Whereas, as soon as the fumigants became, are becoming less allowed. You know, everybody's like, you know, we're gonna die and everything's gonna, you know, break down. We're not gonna be able to farm. But you know, with organic amendments, there's a lot of great research that can address some of those concerns. So, you know, we just have to kind of identify which chemical that the plant releases is the right one, and then try to incorporate that in our mix and see how, you know, we can get that benefit present.
Do you know if there's been any research done with chrysanthemum?
I don't. Do you tell me more about what it's supposed to be?
Do you want to share?
Oh, I use chrysanthemum to repel some leaf hoppers and
above.
lepidophorus.
Okay, yeah, I thought you were going nematodes.
No, not below ground. So.
Yeah.
Oh, okay. Okay.
Yeah.
That, you okay.
Yeah. So they'll. I can't remember which group I talked about now, but yeah. So there's all this, like, you know, communication above ground, and they're releasing these chemicals. We can't smell them, but insects can, and if they're nearby, they'll come in and help ward off that.
They love to multi-crop with flowers.
Yeah. Nice.
How important do you think it is to identify the biological species that are interacting with these cover crops?
As a test that somebody should do on a regular basis, or just in general?
Well, just understanding its
I'm gonna
IMO,
interrupt. Sorry,
Yeah.
I'm a jerk. 2 minutes, everybody. Then we're gonna wrap it up.
or they're doing DNA studies to like identify
Yeah.
who's in there. How
Yeah.
important do you think that is?
I think in the long term it's extremely important. You know, today, right now, for you know, a farmer trying to understand their biology, I think it's something that's a very emerging area. But I can tell you that the technology out there today, they're understanding this functionality, and they're linking specific microbiomes with different plants. And once they understand what that chemical communication between those two, they'll be able to kind of design these perfect systems, not perfect, these, you know, more ideal systems that can create, you know, the nematicide properties, the nutrient cycling, the microbiome that's going to ward off some other disease. Even drought, you know, you can create different microbiomes that's going to help that plant deal with drought conditions as well. And so once they can identify it, then they can kind of build that better system and you kind of create these unique environments to address all the different resource concerns. So I think it's pretty important. Yeah.
I think that's a good question. Couldn't we, though, assume that because one of the criteria of sustainable farming is the number of earthworms per square meter, that if you have a really high number of earthworms, you're by default you're going to have lots of microbes in there if you're doing a polyculture and you're no-till.
So
It's a very general statement. Would you agree with that or not?
well, I have a, you know, earthworms are a kind of an interesting species that where they're non-native, they can actually cause some problems.
We have 11 species here.
You have 11, yeah. So encouraging
All of
those
them are alien.
the native populations, absolutely. They're kind of a
We
key.
have no native populations.
Oh, okay.
They're all invasive. They've all
Yeah,
been
see, so
introduced.
that could be, you know, I don't know, but I don't know if maybe the earthworm then perhaps isn't like your keystone species. Maybe there's another, you know, beetle